Hello, good evening and fond greeting to everyone to our today's webinar on topic why do we get variable efficacy with medicinal plant extract? My name is Vaishnavi Bhaskaran and I'm here to host today's webinar. Before we begin, I'd like to give a short and crisp introduction about our foundation, Organogenic, which is an agri-biotech startup based in Chennai and incubated in Birax, Bionist, Golden Jubilee Biotech Park for Women. Okay, let's begin. In today's fast-paced world, when being a research scholar itself is a titan matter to approach, right? Dr. Nutan Kaushik has contributed a great bouquet of knowledge and research works to the science world. Not only stopping with national level research work, she has crossed the borders to work in international researches, being as a lead in many of the research projects. She has served many honorary positions, which will be a truly welcoming inspiration for upcoming research scholars. Dr. Nutan Kaushik serves as a Director General of Food and Agriculture Foundation at Amity University, Noida. She also holds USDA Cochrane Fellow at Michigan State University, USA, Nuffic Fellow at Wageningen University, Netherlands, DPT Crest Fellow at University of California, and DART Research Ambassador. She has handled more than 40 projects as PI and co-PI and more than 50 publications to her credit. Now, let me begin. And I very much honorable to welcome our speakers. And I would like to request the participants to ask your questions in between the webinar in the chat box so that we may have an ask your doubts session, like questionnaire session at the end of the webinar. So I would like to welcome our speaker, today's speaker, Nutan Kaushik, madam.
Okay. Can I? Thank you. Thank you, Vaishnavi, for such a nice introduction. So good afternoon, everyone. Today, as Vaishnavi has already told you, that I will be talking about why do we get variable efficacy plant extracts. Actually, when Shamini approached me, at that time I was thinking, what should I talk about? Because there's so many topics which I can talk about. So then I thought, since during the COVID time, everybody is talking about the medicinal plants and looking forward towards the medicinal plants. So we should talk about something which is of prime importance and where we should give some kind of priorities and where we should take care when we are talking about the medicinal plant extracts. So in this picture, you see the berberis plant. This is the root of the berberis. This is the tree of the berberis, leaves, and then the wood, and then this is the extract, and this is the berberin which is present in the berberis plant. This is used for the cure of the liver disease. So I'll be talking about that later. I'm not able to change my slides. Ganesh, I'm not able to change my slides.
Ma'am, I think you can do it.
Pardon?
Just escape and join again.
Share again. Share again.
Yes.
Stop share and then share again. Okay. Okay, let me see. Yeah. So before I come to the topic of my presentation, I'd like to introduce my foundation, which is Amity Food and Agriculture Foundation. And Amity University doesn't need any introduction. However, this foundation, I would like, because it's not known to many people, so I just like to introduce this foundation. Amity University, right from the beginning of its inception in 2005, has started the Institute of Organic Agriculture and also the Amity Center for Agriculture Extension, ABG Center for Soil Science and Health. So likewise, there are 8 and 10 departments which are dedicatedly working in the field of food and agriculture. And so our founder president, Dr. Ashok K. Chauhan, this agriculture is very close to his heart. And then he established his foundation for conducting the research leading to the excellence in area of agriculture, veterinary, animal dairy, and fishery sciences, and dissemination of the innovative technologies, imparting education in agriculture and integrated fields, and also developing globally competitive human resources. With this vision, we are working in many areas, and the main uh, there are many areas of research areas, and medicinal and aromatic plant is one of them. So we are working in organic agriculture, post-harvest technology, soil health, agriculture extension. As I told you, we have 8 to 10 research centers, and we have manpower around about 30 to 60 people are working in this area, and many PhD scholars are working. So they are working in various areas, nanotechnology, food technology, and vertical farming, and as well as the metabolomics. So these are the, and gene editing is also, so various across area. So now coming to this medicinal plants, I think everybody knows that about 1200 plant species are used in. We have like our traditional system of medicine, which is Ayurveda, Siddha, and Unani system of medicine, where about 1200 plant species are used as medicine. And about in allopathic system also, 25% of the pesticide pests are sourced from plant, and 7,500 plant species are reported to be used by the tribal community. Tribal communities plants are not part of this Ayurveda, Siddha and Unani system. So if we do the total together, it's more than 10,000 plants which are being used in different system of medicine in India itself. I'm not talking about the Chinese system of medicine, but this is I'm talking about the Indian system of medicine system. So if we in the if you see really see the opportunities as far as the medicinal plants is concerned, is tremendous. As we see, there are 25 biodiversity hotspots identified all over the world, and out of those 25 biodiversity hotspots, 4 are present in India. So we have tremendous biodiversity opportunity which can be explored for getting new molecules. And now, during the COVID-19, the medicine plants has been become has gained again an importance as immunomodulatory source for immunomodulatory properties, and also this as a source for bioactive substances. You see these plants like the cinnamon, curcuma, osmium, piper, which is vinifera, and gingiber. The kara of this one is being promoted by Ayush, and a lot of people are taking this kara. But again, my question comes to again this one when I talk about the variability. Like some people say it is effective, some say it is not effective. Why is that so? I'll talk about that later. Similarly, these are the bioactive sources, substances which has been explored against the COVID-19. This is the review which has been published in Frontiers in Pharmacology in August 2020, and it gives list of the species plant extracts which has been like tested against the COVID-19, and we see that some of them have been found quite active. So all of this is leading. So, what is the threat to the medicine plants? The threat is that because of this demand, unmindful exploitation is creating threat to existing flora and fauna. And so, we because most of them are sourced from the wild habitats, and very few are cultivated. So we need to do the cultivation and restoration of the natural habitats so that they remain as a source for continuous source for the bioactive substances. Now, again, the scope and opportunity is that now we have the renewed interest in traditional system of medicine, and COVID-19 is also one of them, one of the factors leading to this opportunity. And on the other hand, if we really see only 3,000 plant species has been investigated scientifically, there we have about 17,000 plant species reported. Only vascular plant species are reported from India, and out of that, 3,000 are only been investigated scientifically. So there's a huge opportunity that that we might even get new sources for medicines from these plants. Now, now coming to the efficacy of the and safety of the herbal medicine with controlled clinical trial. So when the clinical trials are done, we get variable efficacy with extract. Some extract they work very well, some extract they don't work very well. And what is the reason? The reason is the lack of standardized and quality control of the herbal drugs used in the clinical trials. Anybody can go to the they get the plant either from the market or they get from the some suppliers. Either the species is not right or the what you call the raw material which has been sourced is not the right. It doesn't have the optimum quantity of the active principle. So use of the different doses of herbal medicine is another factor which is leading to the variability in the efficacy. Then inadequate randomization in the most studies and patient batch not properly selected is another reason for this variability. Then difficulty in establishing appropriate placebos because of the taste and aroma. And then last is wide variation in the duration of the treatment using herbal medicines. But above of these, why efficacy varies is because of the uneven doses of the active ingredient present in the extract. The extract which has been used as a for the clinical trials has different efficacies because the active principle which is present might not be the same in the trial 1, trial 2 or trial 3 means like if person 1 is doing the clinical trial maybe sourcing the extract from somewhere else, the second person who is doing the clinical trial we may be sourcing that extract from some other source. So because the extracts are sourced from from different places, from different batches, that might be the reason that so the amount of active principle is present in the extract is directly influenced by the concentration of AI in the raw material. So the raw material used by different people are different. So they are so the active and so the amount of the active principle which is present in this extract is also different. So therefore, determination of concentration of the active ingredient in the raw material is very important. So it is a raw material, the amount of AI which is present in the raw material which determines the efficacy of the extract. If your raw material has the right amount of the AI, it will have it will give the efficacy. But it has a low amount of the AI, it will not give the right efficacy as you are looking forward to. So this is why you get the variable efficacy with the medicinal plant extracts. So why the plant where why there is a variation in the secondary metabolite concentration in the raw material? That's because of the 6 reasons. One is the seasonal variability. The amount of effective principle which is present in the raw material depends, it is either increases or decreases, varies from season to season. In one season, it might be low, in another season, it might get increased, then again, it might get low in the another season. So we need to optimize the season in which we get the maximum amount of the secondary metabolite concentration in the plant. Second is also the physiological stages of the plant, particularly in the case of the tree species. Tea species, the age of the tea species also affect the concentration of the secondary metabolites. The young trees may have lower up, sometimes the young trees have the high amount of the some secondary metabolites. In some cases, it has the low amount of the secondary metabolites. So we also need to optimize at what stage we can we can harvest the raw material at what age or what physiological stage. Sometimes whether it's a reproductive stage or whether it's the other stage where we can non-reproductive stage, which is giving the highest amount of the secondary metabolite. Third is also the geographical reasons. The soil, the temperature, and the soil type also affects, and the altitude affect the amount of the secondary metabolite. Then environmental conditions like the temperature, humidity, all that also affect the amount of the secondary metabolite in the plants. Then harvesting time, as I told you, all this affect the harvesting time. What time you have harvested, like the harvesting time depends on the season and the geographic the season and the physiological stage of the plant. Then genetic factor. Genetically, there may be population variability may exist genetically. So that's called the varieties. We may have different varieties depending upon genetic differences. Genetic differences are the permanent differences. They will behave. So both together, the genetic and all these factors together, if impact the amount of the secondary metabolite which is present in the medicinal plant parts. So to this, that, so all these problems make the bioprospection of the medicinal plant species as a mandatory things. So we must do the bioprospecting of the biological diversity for the secondary metabolites or for the new genes. And this bioprospection for biochemical prospecting is the one which is and is the one which is, I'm talking about. So this could be divided into two types. You can do the prospecting of the plants in two ways. One is with the known active metabolites, where you know that we have already identified the active metabolites like the Artemisia annua, like Vidania, we know the vidonolites. So prospecting of diversity for known is that for in different plant varieties or accessions we know, and here we need to identify then another category is where there are many Ayurvedic plants where traditional knowledge is there for the activity, but we have not identified the active molecules. So there we have to do the prospection for the identification of the active molecules, which is responsible for the activity through the bioassay mediated isolation of the plant extract. So this is the approach we follow for the known active molecule. Like we collect the samples, so we need to do the population variability of the active molecules. So for that, you need to collect the samples from the diverse germplasm of the plant species, quantify the active principles by using with the help of the like GLC or the HPLC, depending upon whether the type of the secondary metabolites. The essentials you can do by GLC, others you can do by HPLC. Identify the elite plants in terms of the active principles. So what are the different active principles, which are the plant giving the highest amount of the active principle can be identified as the elite plant. Then mass multiply or through the tissue culture. So that elite plant becomes a variety. That is the one which we should be used for extraction of the multiplication, extraction of the preparation of the extracts. So, but since the plant is numbers are very few, so initially you can multiply these plants using the tissue culture, and then for the cultivation purposes, and then later on you can go by the vegetative propagation. So this will help the conservation of biodiversity by sustainable harvesting to meet the demand of the raw material. Similarly, sorry. So for unknown, we do the prospection by using the sampling, doing the sampling of the plant and identify the collective species and ascertain the local status of the species. Then we use the biochemist for suitable protocol for sample preparation and standardization of method for GLC and HPLC and quantification of the and so it is a multidisciplinary approach. Then tissue culturist, we need a tissue culturist for development of the protocol and then agronomist for developing the cultivation rules. So you know that active principle determine the market rates. Like Taxol in Texas Pacata, Vidalolites in Vidania somnifera, Azulin in Chamolia, Pythons in Chrysanthemum, Diacetylenine in Diaspora. So these are all species where we do the active principle. If the active principle is high in the plant, the seller of the plant will get the high price. So this is an approach which we follow for bioprospection of the plants with unknown active molecules, where the activity is known traditional by the traditional knowledge, but we need to do the scientific validation of that activity. How do we do that scientific validation? We do the by collecting the plant part. So we need a botanist to identify the plant species first, and also and then we in preparation of the we do the preparation of the extract, we do the bioassay, and then the chemist identify the active principle. Again, the bioassay is done. It is bioassay mediated fractionation, and then once it is identified that we know the active principle, the patent can be filed for that active AI, and then we can do the formulation preparation, toxicological evaluation, screening of the elite plant, and then multiplication and DNA fingerprinting of the elite plant, and then we can go for the commercial exploitation of this one. So if we follow this approach, then the variability of the activity will not come in the extract. So we need to identify the activities for this one depending upon the traditional knowledge, whether it's antiviral, bactericidal, or fungicidal or insecticidal. Then standardize the bioassay need lab. So we need lab secured with the GCMS, HPLCMS, and the NMR and so on for this type of activity. So now coming to the seasonal variations. So seasonal fluctuation have impact on the availability of the active principle in medicinal plants. Hence, therapeutic efficacy also get influenced. So this happens particularly in the case of essential oils. Essential oil components show rhythmic change in oil production and throughout the growing season. Hence, harvesting time need to be standardized for each of the species individually. So in next table, I will, so effect of seasonal variation on volatile components. You can see that in the thymus sulfilum, in the summer season, the content is 0.28%. It is the highest during the summer, while it is the lowest during the autumn and the winter. It is 0.17% too, and it goes as low as 0.07% in winter. Whereas in the case of a mentha, if you really look at the third bullet point, in the mentha camerulensis, the sorry, it is also lowest in May. Menthol content is 5.3% in the winter, and it is low in May, that is 3.5%. Whereas in the case of Osmium basilicum, it shows maximum amount of essential oil in winter and minimum in summer. So each species varies in terms of the amount of essential oil which is present in the according to the season. So it's not that all plants will have high activity during the summertime, or they will have low activity during the summertime, or they will have the high activity during the winter time, or the low activity during the winter time. So for each plant, you need to do the studies and then identify this. Then effect of seasonal variation on non-volatile secondary metabolites. The previous one we talked about so total phenolics in Camellia sinensis has been found to vary according to the season. Like in July, it was 80.69 microgram per milligram, whereas in the September it was 62, and it further decreased to 59 microgram per milligram in May. Similarly, in podophyllatoxin also, it has been observed that leaves of immature, mature male or mature female plants was significantly higher than the leaves of the juvenile plant. I talked about the effect of the physiological stage of the plant. So this particular study shows that the effect of the physiological stage of the plant, where the samples were collected from different physiological stages, and then they found that the female plants, if they are collected in mature male female plants, it gives a high amount of the podophyllatoxin, whereas the leaves of the juvenile plants gives the low amount of the podophyllatoxin. So sampling date also affected the podophyllatoxin content. The leaves harvested in January and April exhibited a higher podophyllatoxin content, and then the leaves harvested in February and June. So depending upon the plant part which you are harvesting for podophyllatoxin extraction, you have to identify the right time for the harvesting. So this is the effect of seasonal variation and in volatile content on some important medicinal plants. You can see for different plants there is a different season, optimum season has been optimized. So you see for this for Artemisia, it is the autumn and summer, and for Cistus, it is a spring. Whereas for Pseudopodium, it is the summer. For Eucalyptus, it's the autumn. For some plants, it is a summer. For some plants, it's a spring. Some plants, it's a winter. So season. So according to the season, the active principle or the volatile oil content has been found optimum at in different seasons. So you need so for each species you need to optimize. If the information is not available in the literature, you have to optimize on your own. So the effect of seasonal variation on the non-volatile compound, again, in the previous slides, we saw the volatile component. These are the secondary metabolites, which are non-volatile in nature. And then again, here also, we found that different metabolites show different trend in terms of the seasons. So some are like the Turamenta, the choline and alkaloid content was high in rainy season. For Bidania, it is the winter season, and then for flavonoids also, it is the winter season. For phenol in Apis mellifera, it is a winter season, whereas in Atatura, it is the rainy season. So for different AI, the time of harvest is different. Uh, this is the continuation of the previous table. And this is the effect of seasonal variation on the pharmacological activities of the medicine plants. In the previous slides, I showed you the variation in the active principle. Some people they also did the efficacy of the extract collected prepared from the plants collected in different season, and then they study the efficacy. So you see here, efficacy also varied. Why the efficacy varied? Because the active principle varied for this for different for the material from which you are preparing the extract. So for if you really see this table, this is the Biotas communis antioxidant activity was highest in rainy season, where all these three, all one, two, three, four plants, all these four plants are having the antioxidant property. However, the activity which is showed highest is different for the season in which they showed the highest activity are different. This plant shows the winter and summer as the highest activity. Again, this plant with summer and winter. This is the winter and spring, and this plant shows the rainy season as the highest. Show the highest activity during the rainy season. So the time of seasonal variations does affect the efficacy of the extract. Similarly, again, it's the same continuation of the same table. And then this is a study which I did for the variability studies in Vidania Saminifera. So we collected 162 accessions from different states of India. We standardized the HPLC method for estimation, and then we did this we also did the polysaccharide estimation of the root. Root contains a lot of polysaccharide in Vidania somnifera. Root contains, and traditionally it is the root which is used for its property, not the leaves. Whereas in the traditional system of medicine, whereas the all the efficacy studies in Western world has been done with the leaves, which is with the witherolites. So chemotyping was done, and then so this is the, if you really see this population variability, so after the collection of the seed, we collected the seeds and then we grow in one particular location to avoid the effect of location on the variability of the active principle. So if you really see the variability, there are different morphotypes. So some the wild ones are the tall type, whereas this the one which you see in the front in the left picture, it is a short type, which is the cultivated. Most of the cultivated varieties, all the cultivated varieties are the short type, whereas the wild types are the all the tall types. So Bidania somnifera has three chemotypes. They are known by literature that it contains three types of chemotype 1, chemotype 2, and chemotype 3. What does it mean? It means that the plants can contain the chemotype 1 has been constituted as a vitaferin A. It means if the plant belongs to the chemotype 1, it will have the extract of that plant will have vitaferin A as a main active principle. It will not have withanolide. Whereas if if you're preparing the extract from chemotype 2, which contains the main constituents and withanolide D, will have withanolide D. Vitelloferrin A and vitelloferrin D will have different properties, what you call pharmacological properties. They do not have the similar pharmacological properties. So if you are collecting preparing the extract from chemotype 2 and not from chemotype 1, then the efficacy will be different. Similarly, if we see the chemotype 2, it is an intermediate, it has the methanolite E as the main constituent. So if you are collecting the plant, which is the chemotype 3, and preparing the extract from the chemotype 3, third chemotype, then it will have, and you are, whereas the efficacy which you are looking forward to is from the vidaferin A. So it will not give the same efficacy as the vidaferin A because it is a chemotype 3, it is not the chemotype 1. So you must ascertain your chemotype which you are collecting from the plant. So you must identify the chemotype before you go for the collection of the plant. So when we did the HPLC analysis of this 162 accessions, we found that Indian chemotype collection belongs to the chemotype 1. Uh, so this collection from Mandsaur, Yamuna and Noni, they belong to the chemotype 1, having the vidaferin A as the main constituent. Whereas the other collections which are collected from Bhopal, Gopeshwar, Jaipur, Delhi, Jodhpur, Udaipur and Chandigarh regions, they were the intermediate type of the chemotype 1 and chemotype 2 by having the both the peaks vidaferin A as well as the vidaferin D. So it is not the mixture, it is the intermediate of the chemotype 1 and chemotype 2. So it does not have even the single peak. So this is the chemotype 1 having the vidaferin A as a main constituent. So then we also studied the effect of growth stages on the vidanolite content, and you really see this is the different season. These are the three accessions. So here in this picture, in this graph, you see the accession wise variability. This accessions having the high vidaferin content, whereas this accession has the lowest vidaferin A content. This has the intermediate vidaferin A content. But and this also shows the seasonal variability. This sections, all the three accessions were collected during November, January, and the March. So highest content was during the November, and the lowest content was during the March, where that is just before the harvesting. Harvesting is done in somewhere in April and May. So the vidaferin content is highest during the vegetative phase and lowest during the harvesting is maturity stage. This is the same trend in all the three accessions. So trend remained the same in the accessions. However, the amount is varies from accession to accession. So trend, it is not the trend which is varying. Trend doesn't. So the harvesting time of a species remains the same for all the population. Whereas, however, the active principle variability will vary from plant to plant, from population to population. So this is again, you see, this is the 6 days, this is the average content of all the accessions, 6 days after after planting, then 6 days, this is the leaf, this is the 180 days after planting the leaf, and this is the root containing 60 days after the planting, and this is the root after 60 days. Why we estimated in both? Because initially we did only the analysis on the leaf, and we thought maybe the vidaferin A content is getting translocated at the time of maturity into the root system. So let's see how much is the vidaferin A which is present in the root system. But what if we release? But we didn't find the translocation of the vidaferin A from root to the from leaf to the root, because in the root also at the time of maturity the vidaferin will also decrease. So and then we so if we really look at this table, if we really look at this table, this shows that the minimum and the maximum, the variability in the population. So we got as low as 0.22 percent, 0.2%, and as high as 3.3%. Just imagine, I extract coming from the 3.3% Vidaferin A containing plant and the extract coming from 0.2% Vidaferin A containing plant. Can you imagine? The efficacy will certainly be higher for the extract which is coming from 3.3% containing plant than the 0.2% Vidaferin A containing plant. Okay. Similarly, here the efficacy goes further down. Is further down in the root. Root is as low as 0.003%, where the maximum was 0.75%. Similarly, in 150 days old leaves, the Vidaferin content is 1.1% and 1.2%. So the Vidaferin A content initially was low, and it further increased at 150 days, and it further decreased at 180 days. So it means if you're collecting the extract at this stage or this stage or this stage, the variable it will give different efficacies. Same plant but collected in different seasons, different time will give different efficacy. Why? Because the Vidaferin A content is varying at different seasons. So this table shows and like and also not only shows same plant uh, but within the same season also, if you're collecting from different populations, the variability will differ. The efficacy will differ because different population have different Vidaferin A content. This is the polysaccharide variability you can see in the different accessions. So all accessions, this polysaccharide content was done at the time of maturity because the highest content was in at the time of maturity. You see this particular accession has as high as 45% polysaccharide content. And this accession has only 5%. If your active principle is polysaccharide, then what do? If we are collecting these accessions, which is contained only 5%, you will not get the efficacy. But if your active principle is if you are collecting this plant, which is contains the 45%, the efficacy will be very high. Similarly, this is the study which we did with berberis. So berberin is the active principle which is present in the berberis plant. So we did, and it is used for liver problem for hepatitis. It is present in the lip 52. It is like one of the ingredient of the lip 52, which is a very popular Ayurvedic medicine. So here we see the variability 1.6, 0.29% and 0.098%. So again, you need to identify. So this bit again shows that different locations, the variability is different. The Vidaferin A content is like representative in blue bar. So it is and then the red bar. So it is 13. It is goes as high as 13% in the extract, and then 12% and 8.4 and 9.9% in the extract. So this shows in different species. Like all the species looks like the same. So if you're collecting the wrong species, then also your efficacy will be different. So this I will skip. And this is the case of the neem, which we studied. Neem contains a lot of active principle. Neem leaves are like azadirachtin content 0.02. We found that it varies in the leaves from 0.02 to 1.6%. So if you're collecting the neem seeds, which is from containing the 0.02, you will not get the efficacy. Whereas this particular extraction will give the efficacy. And we also found, we collected from entire India, and we found the seeds coming from the south contains the high azadirachtin content, whereas the seeds coming from the north contains the high oil content. There was inverse relationship between the oil content and the azadirachtin content. Azadirachtin is present in the de-oil cake, whereas the oil is after extraction of oil, you get the azadirachtin content. So this content was highest So if the industry is looking forward to the oil content, then they should go collect from the northern state of the part of the India. If they're looking for the azotite content, they should go for the southern state of the India for collecting the material. So not and then this is the variability which is present in terms of the different fatty acids which are present in different accessions. Oleic acid varied from 32 to 68%, palmitic acid 6 to 46%, linoleic acid 3 to 36%, stearic from 0.05 to 26%. So this is the variability which exists in the population. So to conclude, I would say that it is important to study the population variability of the active ingredient and identify the right variety, right geographical location, and right time of harvesting. This is very important if you really don't want to get the efficacy in your extract. Then standardized extract must be developed for all medicinal plants to get the clinical efficacy. So in the end, I would like to say that scientists may be able to make any molecule that they imagine on a computer, but Mother Nature is an infinity more ingenious and exciting chemist. You so there's a possibility that you get every time you explore the plant, you get a new molecule. So with these words, I would like to say thank you and look forward to the question and answer session. Thank you.
Thank you so much, ma'am. That was really explanatory and informational to all the scholars. And we would like to go to the question sessions. So we have a question from Rashmi. Um, is the variation in metabolites of plants also observed in the plants which are grown in polyhouses under controlled conditions?
Yes, because if the variety is the same, ideally it should remain the same. But if we are using, if the seed is not a standardized seed, if it is a mixture of different populations, you will we will be able to see the variability. But variability will only be in terms of the seasons, different seasons. But population variability should not be there.
We hope that answers your question, Rashmi. And we have one more question from Dr. Amarnath Singh. What is the effect of seasonal variation on quality and quantity of alkaloid contents in medicinal plants?
As I told that for each plant, we need to standardize the time of harvest. The alkaloid content, like in case of berberis, what we saw that the vegetative stage is the right time for the alkaloid content. Berberin is an alkaloid. Okay, but whereas in the case of Vidania also, which is an not an alkaloid, that's a vidadolide. So, but in that Vidania also we saw that the vegetative stage is the right stage. But as I showed you in the table, that for different plants, for different activities, for different even the for alkaloids also, for some plant it was the winter season was the right season. In some plants it was the summer season which was the right season. So is depending upon the species and depending upon the metabolites which you are looking. One plant may contain 2-3 metabolites also. So which alkaloid you are looking forward to? It is a berberin or it is something else which you are looking forward to. So you need to optimize your harvest time accordingly.
Thank you, ma'am. And we have another question from the same person. How the effect of seasonal variation takes place in medicinal plants?
Seasonal variation, how does it work? It's because of the gene expression basically. The genes are responsible for production of these secondary metabolites. So expression of these genes are controlled by the like the temperature and the humidity and difference, and then the light and then the photo period. So all these factors and leads to the expression of these genes, and depending upon the expression of these genes, the amount of secondary metabolites also vary.
We hope that answers your question. And a question from Ashwagandha. What is the chemical constituents of Withania somnifera?
As I told you, the Vidania somnifera contains 3 chemotypes. So, like particularly the Indian population contains vidaferin A as a main constituent, and some plants, as I showed you, belong had the pyrin A and Withanolide D. Although others are also present, but not as the major constituents. These are the major constituents of the Indian population. But if we go to the Israel, Israel contains withanolide A, withanolide E also. They have the chemotype 3 also. So these are the different chemotypes which are present in different populations. Yeah.
Thank you, ma'am. And there is a question from Ankur. Sorry, Ashwagandha also suits you, maybe. Ashwagandha's chemical component name was Withania somnifera. Okay, let me go to next question from Amarnath Singh again. Is there any correlation between growth, performance, and alkaloid content in medicinal plants?
Growth, performance, and alkaloid content. I'm not aware of that because growth has never been correlated with the active principle. But yes, if the biomass is more, you will harvest more active principle because growth is directly related to the biomass. Biomass is what the leaf. If you're collecting the leaf weight will be more if the growth is good, and if the leaf weight is more, you will collect more of the active principle. But unit expression will not vary.
Okay. Thank you, ma'am. And we have a question from Rutella. Can we apply same experimental processes for garden plants with their wild relatives?
Yes, we can. Yes, we can.
So we have so many questions from the participants. So how was your experience these many days, ma'am? I mean, let me jump into general questions. Let the participants think for some time to ask the questions. I mean, how was your research journey all over these years? I mean, you have a 25 years of experience in the research field. So how was it? What are the different? I mean, in some of the day, you could have had a thought like, yeah, I have changed this thing, which could be the contribution to everything. What you have done is a contribution to world science world. But even though for you personally, some of the thing which you have made or developed would have been inspired yourself. So is there any experience of that, ma'am, so that we can hear about it?
My, my journey, research journey. Okay. I started my journey. I did my PhD in agriculture chemicals in pesticides actually. So that was pesticide residue analysis. So for residue analysis, you are exposed to, you need to do the analysis of the pesticides. So you need to handle the GC and HPLC. There I got exposure to the analytical chemistry. And during my postdoc, I did a project on Neem, variability of the Neem plants. And there I got
Has amazing medicinal values.
So that was there I got exposed to the natural products. So that's how I got into the natural product chemistry also. And then after that, I joined Terry, and there I did lot. I joined the Brassica project. There I learned I did this breeding of. I joined the breeding group. So they were doing the breeding of the double low hybrid, double low varieties of the double of the Brassicas. So my job was to give them uh, the identification of the plants with the low uric acid content and the low glucosamine content by using my analytical expertise. And it was that we need to screen thousands of germplasm within 2 months. So it means a really very efficient method because the method which was available at that time for fatty acid analysis, like you had the GC method, was used to take around 45 minutes for 1 month. And we didn't have the auto sampler also. It means in a day we can do only 5 to 6 samples and that's it. But my demand was to have at least 40 samples in a day, as per the number of samples which we had. So I had to optimize. So I designed a column, packed column. My GC had the packed column. We didn't even have the capillary column. So having the low runtime for the packed column, uh, so I did some literature survey, and based on that, I asked the Nutan Mr. Batra. I contacted Mr. Batra, and I asked him, "Can you pack a column for me?" And he was very happy, and then he did pack the column for me, and the runtime was fantastic. The skinning can be done in 5 minutes, and the composition can be done in 10 minutes. So so that was the contribution. So I'm just telling the younger generation, do not think that this is not possible. Everything is possible if you think carefully. So try not to get bound yourself with whatever is available. You think how to make the things, you make your own target and try to work according to that target. So that was the first thing which I did. Second thing was that in Western world, they used to do this glucosinate analysis by GLC, by doing the derivation. Uh, because glucosinate contains, because it is a it has its sugar and also the sulfonated compounds. So they used to do this derivation, and this derivation agent was very costly. I said I will do by HPLC, the intact glucosinate. So I stood by that method also, doing the intact glucosinate analysis without derivation and by using, just by modifying the mobile phase. I use a modifier and which used to separate, because they, being the anionic in nature, cannot be separated on the column. They come all together. So what I did, I suppressed the ion, charged ion, by using the, because it has a sulfur negative. So I use ammonia as a suppressor. So they form the group, and then they could be separated on the reverse phase column, normal column, on the normal column. So that's why I did. So that's how I did method of intact analysis of glucosilate. So we could use our own HPLC for doing the glucosilate analysis. Then later on, then I started working on biopesticides and so on. Then my first collaboration was with Germany, with the University of Düsseldorf. And
Yes.
afterwards, I did a lot of international projects, and I did. Then I went to then I did Indo-French project, then Indo-Australia, Indo-US, Indo-Portugal, and then this Indo-Australia, and then recent
It's really an
Indo-North.
inspirational journey, actually, to all of us. That's
So,
why I would
so,
like to
so,
know from
so
you.
once you start doing the network gets expense on your own. So it's not that. So I'm just telling the students that please do not think something new. Put harder targets for yourself so that you achieve whatever you want to achieve, and
It
then
applies for every field, actually, every
everything.
person's in this life. I mean, whoever is very passionate on their career.
Yes. So this is, and now I have become so again back to agriculture. In agriculture also, I'm just looking after many things. So natural products, biopesticide, and so on. So it's everything new as a challenge, and something
Mm.
which you can learn, give a new
Yeah,
learn.
true.
Not as a, this is not my area, I can't do. Don't think
Yes.
that way. You should take everything new as a new challenge and try to achieve, learn, read about it, so and then do it.
Yes. Thank you so much, Mom. Thank you so much. So, we have a question from Dr. VK Singh. Is there any global compendium of volatile substances of
Yes,
medicinal plants?
yes, there is. In If you go to there's a website. In fact, there's a website where you can get all the essential oil composition of the different oils. Then NIST library is also available online. If you type, you can get the composition in NIST library also. There are GCMS libraries which are available and which can give you the composition of the essential oils. And there are compendium also which are available.
We hope that answers your question, Dr. VK Singh. And we have another question from Bhaskar Chowdhury. Is synergistic effect appear in all mixed formulation of plant drugs?
Yes, you get the synergistic effect of the different components which are present over there. So that's why we use the crude extract. We don't use the active principle. So when you are doing the efficacy, you are doing the efficacy of the crude extract, not of the pure compound. Pure compound you do later on, just to prove that this is the active compound which is present in it. It may not be it is the most active compound, but it may not be as active as your crude extract, because food extract has other many other compounds also which are giving the working together synergistically. This I have seen myself, like the pure compound is not as active as the your crude extract. And that's because, and using the crude extract, there's another advantage of using the crude extract, that the buildup of the resistance is not so fast. In case of pure compound, the pathogen or the can develop the resistance very fast because it is a single molecule. So yeah, so detoxification is very easy. Whereas in the case of food extract, it's a multiple compounds, 50, 60, or sometimes even 70 compounds are present over there. They may not be active, but the compounds are there. So developing the resistance in the for the food extract for the pathogen will take years and years together. Maybe take 15 years or 20 years to develop the resistance. Whereas in the case of pure compound, it may be within 2 years you get the resistance. So besides the synergistic activity, you also overcome the resistance problem, build up of the resistance problem.
Thank you so much, ma'am. And I guess now we have come to an end of this session. So I request the participant to kindly fill in the feedback form, which will be available till 12 o'clock today. And I thank Dr. Nutan Kaushik for giving us a factual and educative webinar. I would like to thank Dr. D. Sudhakaran, GM of Golden Jubilee Biotech Park for Women Society, for his support, and Dr. Shamni for making this session possible, and thank our mentor, Dr. Ganesh Mani, for being extremely supportive for each and every sessions conducted by Organogenic. A big thanks to all the participants and hoping your presence in the upcoming sessions. And this webinar session will be recorded and being uploaded in the YouTube video, and you can view anytime you wish. And please subscribe and follow us in the YouTube channel too. Thank you.
Thank you. Thank you, everyone.